DNA replication. The synthesis of new DNA molecule from pre-existing DNA is called DNA replication. Each DNA molecule produces exact copies of its own structure by replication. The common method is called semi-conservative and was proposed by Watson and Crick. In this method, each strand of the DNA molecule acts as a template for the synthesis of a new complementary strand. As one strand of the parent molecule is conserved in the daughter molecule, the method of DNA replication is called semi-conservative method of DNA replication. This picture explains the semi-conservative method of DNA replication. The main enzyme required is DNA-dependent DNA polymerase enzyme, since it uses a DNA template to catalyze the polymerization. These enzymes are highly efficient as they have to catalyze polymerization of a large number of nucleotides in a very short time. They also catalyze the reaction with high degree of accuracy. Deoxy ribonucleoside triphosphates serve dual purposes. They act as substrate as well as they provide energy for polymerization reaction. In addition to DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. Here listen, any mistake during replication would result in mutation. Energetically, replication is a very expensive process also. The replication occurs within a small opening of the DNA helix referred to as replication fork. Here you can see the replication fork structure. The replication occurs within a small opening called replication fork. The DNA polymerase enzyme catalyzes the polymerization reaction only in one direction, that is 5'3'. Therefore, in one strand, the replication is continuous and the other it is discontinuous, and the discontinuously synthesized fragments are joined by the enzyme DNA ligase. There is a definite region in Escherichia coli DNA where the replication originates, and such regions are called origin of replication. So the discontinuously synthesized fragments, commonly called Okazaki fragments, are later joined by the enzyme DNA ligase, as I mentioned earlier. Here you can see the template DNA, continuous and discontinuous strands. Everything explained in this replication fork diagram. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle, and the replication of DNA and cell division cycle should be highly coordinated.